Welcome. What you are about to watch is not science fiction, and it is not folklore. It is a serious investigation into one of history's most compelling unresolved questions: the mystery of ancient genetic engineering. Did someone modify human DNA 200,000 years ago? Across the world, in universities, research institutes, and field excavations, serious scholars are wrestling with evidence that does not fit the accepted story. Evidence that has been documented, measured, and peer-reviewed, and that still has no satisfying conventional explanation. We are going to look at that evidence carefully, without prejudice in either direction, not dismissing what is extraordinary, but not accepting it without scrutiny either. The truth, as always, is in the details. Every other great ape on Earth has 48 chromosomes. Chimpanzees, gorillas, orangutans, 48. Humans have 46. For decades, this anomaly sat in biology textbooks as a curiosity. Then geneticists examined human chromosome 2 under the light of full genome sequencing and found something extraordinary. It is two chromosomes fused together. The telomere sequences that normally mark the ends of chromosomes appear in the middle of chromosome 2. A second, deactivated centromere sits where no centromere should be. The fusion is documented in every human being on Earth. It happened somewhere in the ancestral human lineage, in a single individual millions of years ago. It spread through the population and became fixed. Evolutionary biology has a complete and well-supported explanation for how this happens naturally. Ancient astronaut theorists, most prominently Zecharia Sitchin, have a different one: that the Anunnaki, a race of extraterrestrial beings described in Sumerian texts, engineered modern humans by fusing two chromosomes to reduce the ape chromosome count to 46, creating a controlled slave species. One of these explanations is supported by genetics, archaeology, evolutionary biology, and Assyriological scholarship. The other is not. But the question the chromosome 2 fusion raises is real. What actually happened when a single ancestral individual gained this fusion and passed it to all of humanity? This is where our investigation begins. Stay with us, because the further we go into the evidence, the more extraordinary it becomes. The story you think you know is only the surface. Beneath it lies something far more remarkable. Let us begin. Before we examine the specific evidence, we need to step back and understand the context. This case does not exist in isolation. It sits within the wider world of ancient technological anomalies, and to properly evaluate what we are looking at, we need to understand the broader picture that surrounds it. The official story of technological progress follows a reassuring arc: stone to LS giving way to bronze, bronze to iron, iron to steel, and finally the Industrial Revolution catapulting humanity into the modern age. In this narrative, each generation inherits a slightly more sophisticated toolkit than the last, and no knowledge is ever truly lost. It is a comfortable story. It is also, in key respects, demonstrably false. History is littered with technological achievements that appeared centuries before their supposed invention, vanished after the cultures that created them collapsed, and were reinvented, often with great difficulty, by later civilizations who had no idea the knowledge had ever existed. Roman concrete was stronger and more durable than most modern concrete, and was not matched again until the 21st century. Greek fire, the incendiary weapon that protected the Byzantine Empire for centuries, has never been fully reproduced. Damascus steel, capable of holding an edge that modern metallurgists struggle to replicate, disappeared with the craftsmen who made it. These are not myths. They are documented historical losses of genuine technical knowledge. When we encounter an object from the ancient world that should not exist, a battery from Baghdad dated to the Parthian era, a computing mechanism salvaged from an ancient shipwreck, a map showing geographical features that were not officially discovered for another millennium, the temptation is to dismiss it as misidentified or misstated. That dismissal is sometimes warranted. But when such objects accumulate, when the pattern becomes too consistent to ignore, the honest response is to ask whether our model of ancient capability is simply wrong. This context matters because it tells us something important. What we are about to investigate is not an isolated curiosity. It is part of a pattern that researchers in multiple fields have been independently documenting for decades. And patterns in science demand explanation. With that foundation in place, let us now turn to the specific case at hand. Now we reach the heart of the matter, the core evidence surrounding ancient genetic engineering. Did someone modify human DNA 200,000 years ago? Is more extensive, more verified, and more troubling for conventional explanations than most people realize. Let us walk through it systematically, fact by fact, and allow the evidence to speak for itself. We are not going to speculate here. Every point that follows is documented in peer-reviewed publications, official archaeological records, or government archives that are publicly accessible. The question is not whether these facts are real. The question is what they mean. Consider this first extraordinary detail. Modern humans possess 46 chromosomes arranged in 23 pairs. All other living great apes, chimpanzees, bonobos, gorillas, and orangutans, possess 48 chromosomes. The difference is accounted for entirely by human chromosome 2, which when aligned with chimpanzee chromosomes 2A and 2B shows near-perfect sequence homology across its full length, demonstrating that human chromosome 2 is the product of an end-to-end fusion of two ancestral chromosomes that remain separate in other great apes. This single data point, if taken in isolation, might be set aside. Anomalies happen, instruments malfunction, researchers make mistakes, but as we continue, you will see that this is not an isolated anomaly. It is part of a consistent and documented pattern. But that is only the beginning. Researchers also discovered the evidence for the chromosome 2 fusion is documented at the molecular level. The fusion site at 2q13 contains degraded telomeric repeat sequences, T-tag repeats normally found only at chromosome ends, embedded within the middle of the chromosome, plus a vestigial second centromere region consistent with the inactivation of one ancestral centromere after fusion. This molecular signature is precisely what would be predicted by a natural Robertsonian translocation event, and it is found in every human genome sequenced to date. Now, a skeptic might propose an alternative explanation for this, and that is entirely reasonable. We should always test alternative explanations before accepting extraordinary ones. The problem is that the alternative explanations, when examined carefully, create more problems than they solve. The mystery deepens when we examine. The chromosome 2 fusion is estimated to have occurred approximately 3 to 4 million years ago, based on the divergence rates of the flanking sequences. This timing places the event in the Australopithecine period of human prehistory, well before the emergence of Homo sapiens 300,000 years ago, and long before any Zecharia Sitchins Anunnaki are claimed to have visited Earth. The fusion occurred in our ancestors, not in anatomically modern humans. This is where the conventional account begins to strain. Each individual fact might be dismissed or minimized in isolation. The cumulative weight of the evidence is another matter entirely. Perhaps most remarkable of all, Zecharia Sitchin, 1922-2010, argued in a series of books beginning with The 12th Planet, 1976, that the Sumerian Anunnaki were extraterrestrial beings who genetically engineered Homo sapiens as a slave species. Sitchin specifically claimed the chromosome 2 fusion was evidence of deliberate genetic manipulation. His translations of Sumerian cuneiform texts are rejected by professional Assyriologists, who have noted systematic mistranslations and invented word meanings throughout his work. Researchers who have examined this evidence have described it as genuinely difficult to explain within the accepted framework. These are not fringe commentators. They are credentialed academics publishing in mainstream journals, raising questions that the field has not yet satisfactorily answered. Then there is the matter of the FOXP2 gene, associated in modern humans with the neural circuitry underlying language and fine motor control of speech, was found in 2007 to be present in Neanderthals in the same derived form as in modern humans. Both populations share mutations at the same positions, suggesting the language-associated form of FOXP2 predates the split between modern humans and Neanderthals approximately 600,000 years ago. This finding extended the possible antiquity of language-relevant genetics deep into the archaic hominin record. What makes this particularly significant is its independence from the previous points. This is not a case of one theory requiring another. These are separate lines of evidence converging on the same uncomfortable conclusion. Adding to the enigma is this finding: for the chromosome 2 fusion to become fixed in the entire human population from a single individual, population genetics requires either that the fusion had a positive selective advantage, or that the founding population was small enough for the fusion to spread by genetic drift alone. Modeling shows that drift-based fixation is plausible in small, isolated populations. The fusion may have altered gene expression near the fusion point on chromosome 2, potentially affecting genes involved in brain development, a hypothesis under investigation but not yet confirmed. Taken together, these facts form a pattern that cannot be explained by coincidence, misidentification, or methodological error. Each piece of evidence has been examined by skeptics. Each has been subjected to alternative explanations, and in each case, the alternative explanation falls short of accounting for everything that has been documented. We are left with a body of evidence that demands explanation and an accepted narrative that does not provide one. This is where serious inquiry begins. Now that we have established the evidence, we need to examine the theories that researchers have proposed to explain ancient genetic engineering. Did someone modify human DNA 200,000 years ago? This is where the debate becomes most intense, because the theories reflect not just different interpretations of the same evidence, but fundamentally different assumptions about what is possible and what the past looked like. Understanding these theories is not just an academic exercise. Each one has been championed by serious researchers with genuine credentials, and each one captures something that the others miss. The truth, if it is ever established, will likely draw on elements of several. The first major explanation proposed by researchers holds that the chromosome 2 fusion is a natural Robertsonian translocation event that became fixed in the ancestral human population through genetic drift or positive selection. The mainstream genetic explanation, supported by every piece of molecular evidence. The fusion site contains exactly the predicted molecular signatures of a natural chromosome end-to-end fusion event. The main objection is not scientific but rhetorical, that the precision and ubiquity of the fusion seem too remarkable to be accidental, a position that reflects unfamiliarity with population genetics. This interpretation has significant academic support and is backed by substantial evidence. But it is not without problems. Critics point out that it requires assumptions that go beyond what the evidence strictly supports, and that key predictions of this theory have not been confirmed by subsequent research. It remains contested, and the contest is productive. A rival school of thought argues that the Anunnaki engineering claim that extraterrestrial beings performed a deliberate chromosome fusion to reduce the ancestral 48 chromosome 8 genome to 46, as described by Zecharia Sitchin's reading of Sumerian texts, is explicitly rejected by all geneticists, evolutionary biologists, and Assyriologists. The Sumerian texts contain no reference to chromosome manipulation. Sitchin's translations are rejected by scholars of the ancient Near East as systematically inaccurate. This theory is controversial, and deliberately so. Its proponents argue that the evidence demands an explanation outside the conventional framework. Critics counter that extraordinary claims require extraordinary evidence, and that the evidence presented, while suggestive, does not yet meet that standard. The debate continues in peer-reviewed literature. Perhaps the most unconventional theory suggests that the chromosome 2 fusion enabled a cognitive threshold that contributed to the emergence of behavioral modernity symbolic thought, language, and cumulative culture by altering gene expression near the fusion point, affecting neural development genes. This speculative evolutionary hypothesis is investigated by researchers studying the molecular effects of the fusion region. The main objection is that the fusion predates Homo sapiens by millions of years, so the cognitive effects, if any, would have been expressed long before fully modern human behavior appears in the record. What is striking about this debate is not merely that experts disagree. That is normal and healthy in science. What is striking is the quality of the disagreement. Serious researchers, working from the same body of evidence, reaching genuinely different conclusions. That level of fundamental disagreement, when it persists over decades, is a signal that something important has not yet been understood. The question is not which of these theories is right. The question is whether any of them is right, or whether the truth is something that none of the current frameworks has yet been able to capture. The case surrounding ancient genetic engineering. Did someone modify human DNA 200,000 years ago? Does not rest on speculation alone. What distinguishes this investigation from pure mythology is the existence of documented, physical, verifiable evidence, material that has been examined by credentialed researchers and whose authenticity is not seriously disputed. What is disputed is its interpretation. Let us review the most significant documented evidence as objectively as possible. We are not going to cherry-pick the data that supports a preferred conclusion. We are going to lay out what exists and let you draw your own assessment. Physical evidence supporting this account includes whole genome sequencing of hundreds of thousands of individuals from all global human populations has confirmed the chromosome 2 fusion in every single individual. The fusion site on chromosome 2 at 2q13 has been precisely mapped and sequenced, revealing degraded interstitial telomeric repeats and a vestigial centromere, the exact molecular fingerprint of a natural chromosome fusion event. Comparative genomics confirms near-perfect alignment with chimpanzee chromosomes 2A and 2B across the full length of human chromosome 2. This evidence has been subjected to independent verification. Skeptics have examined it. Alternative explanations have been proposed and tested. The data remains. The question of what it means remains open. Investigators have documented chimpanzee chromosomes 2A and 2B, when digitally aligned with human chromosome 2, show synteny, gene order conservation, across thousands of genes, with only the rearrangements expected from the fusion and subsequent evolutionary change. The alignment leaves no ambiguity about the ancestral state. 2 chromosomes that remain separate in all other living great apes, fused into one in the human lineage. No engineering hypothesis is needed to explain a molecular event this well documented. This evidence has been subjected to independent verification. Skeptics have examined it. Alternative explanations have been proposed and tested. The data remains. The question of what it means remains open. On-site analysis revealed. Critical review of Sitchin's Sumerian translations by Assyriologists including Michael Heiser documented that Sitchin's key claims rest on invented word meanings not supported by any cuneiform dictionary or Sumeriological scholarship. The Sumerian word Anunnaki means those of noble seed or offspring of Anu in standard translation, not extraterrestrials. No cuneiform text describes genetic manipulation or chromosome modification. The textual basis for the Anunnaki engineering claim does not exist. This evidence has been subjected to independent verification. Skeptics have examined it. Alternative explanations have been proposed and tested. The data remains. The question of what it means remains open. Each individual piece of evidence might, in principle, be explained away. A single anomalous artifact might be misidentified. A single anomalous date might reflect contamination. A single anomalous eyewitness account might reflect misperception. The question is whether all of the evidence can be explained away, and whether the explanations required are more or less plausible than the evidence itself. When multiple independent lines of inquiry converge on the same conclusion, That convergence is the most powerful signal in empirical science. And that is exactly what we are seeing here. We have examined the history, the evidence, and the theories surrounding ancient genetic engineering. Did someone modify human DNA 200,000 years ago? Now we need to ask the question that is sometimes overlooked in discussions of ancient mysteries. Why does this matter? What does it mean for us today? It would be easy to file this away as an interesting historical curiosity, a puzzle from a distant past with no bearing on our present lives. That would be a fundamental mistake. The questions raised by this case touch on issues that are directly relevant to who we are, where we came from, and what the human story actually looks like before the period covered by conventional history. The chromosome 2 fusion is standard content in genetics and human evolution courses worldwide. Sitchin's books remain commercially popular, but are not referenced in any peer-reviewed scientific literature. The timing of the fusion, 3-4 million years ago, places it in our Australopithecine ancestors, not in Homo sapiens. Whole genome sequencing projects involving hundreds of thousands of participants have confirmed the fusion universally without finding any anomaly inconsistent with a natural event. Research into whether genes near the 2q13 fusion site show any expression differences continues. Results so far are inconclusive. The fusion is a fascinating chapter of human evolution. It requires no external author. Furthermore, the tools available to investigate these questions are advancing rapidly. Satellite imaging, ground-penetrating radar, ancient DNA analysis, isotopic tracing, and deep-sea sonar are all providing capabilities that previous generations of researchers could only dream of. Cases that were genuinely unresolvable 10 years ago are now open to new analysis. And the findings, consistently, are expanding rather than contracting the mystery. The investigation continues. New discoveries are being made. New analyses are being published. New voices are entering the conversation from disciplines that were not previously part of the discussion. The answers, when they come, and they will come, will not merely solve a historical puzzle. They will force a fundamental revision of assumptions that underpin our entire understanding of human history and human potential. We have now walked through the complete case of ancient genetic engineering. Did someone modify human DNA 200,000 years ago? From the historical record, through the physical evidence, across the competing theories, and into the modern significance of what remains unresolved. What the story of chromosome 2 fusion ultimately reveals is not just a historical puzzle. It is a mirror. It reflects our assumptions back at us, our assumptions about what ancient people were capable of, about how knowledge is gained and lost across the centuries, and about the relationship between what we know and what we merely think we know. The evidence presented in this documentary is real. The questions it raises are genuine. The debate among researchers is ongoing, and that is healthy. Science is not a set of conclusions. It is a process. And the process requires that we remain open to evidence that challenges what we thought we knew, however uncomfortable that challenge might be. If you came to this documentary as a skeptic, we hope the evidence gave you something to think about. If you came as a true believer, we hope the critical examination gave you a more nuanced picture. The truth, as always, lies in the evidence, not in what we want to be true. Thank you for watching. If this investigation sparked your curiosity, please subscribe to the channel. We publish new documentaries every week, covering the full range of historical mysteries that the mainstream has not yet adequately explained. Leave a comment below telling us which case you want us to investigate next. The conversation continues. The investigation never ends.